Hey, my name is Scott,
And Kristen.
and we are the fun team. We're going to go over our week 13 problems and how we got the answers to them. So the first question, it was talking about DNA sequencing, and it wanted us to determine the nucleotide sequence of the DNA molecule shown here, 5 to 3. And so then it asks us to make a banding pattern, which we show here. So this is the original sequence, and so the adjacent sequence would be the opposite base pairs. And so those are the ones that we're going to have on the gel. And so as you see, A coincides with DD-ATP, T coincides with DD-TTP, and so on and so forth. And so the D-deoxy ribonucleic acids, or the DD, whatever they're called, they're going to attach and stop the base at certain points. And so here you get to see where they stopped on the nucleotide sequence, and you're able to see exactly where the where the sequence was, or like how the sequence goes from there. And so that's how we were able to figure out the first part of this question.
Okay, part B says, suppose you use DNA polymerase 1 instead of the Taq polymerase. And if you do that, your polymerase is going to denature and the reactions won't take place because the Taq polymerase is special from a special bacteria that is resistant to heat. So, yeah.
Question 2 asks about adding E. coli DNA helicase to the reaction. So helicase actually isn't needed for PCR. Helicase is mostly used in DNA replication and RNA synthesis, but it's not needed for PCR. So because of that, there's actually not going to be a change in PCR because helicase isn't a component of it.
Okay. Part D says, suppose you added DD-ATP to one of your 4 test tubes as intended. However, you forgot to add the DD-GTP, DD-TTP, and DD-CTP. So if you do that, you'd only see your DD-ATP bands. Nothing else would be there.
Cool. So for the next question, it's all fill in the blank stuff. And so I'll just read these off. The answers are up there as well. So the first one says, during a typical PCR cycle, the synthesizing step is done at 32 degrees Celsius, while the annealing step is done at 55 degrees Celsius. B, oh, sorry.
Okay. B is ampicillin R. AMPR is going to be the common selectable marker that's found in many plasmid vectors, while the restriction enzymes are going to recognize and cleave DNA molecules at the palindromic sites.
C, during agarose gel electrophoresis, DNA is placed in an electric field. DNA migrates towards the answer is positive electrode during electrophoresis, and that's because of the negative phosphate backbone on DNA.
DD-ATP lacks the 2' and also the 3' OH groups.
E, I said transformation is a term used to describe the process where bacteria take up DNA molecules from the environment.
And then F, during the blue-white screening, we're going to use IPTG to induce the expression of the lac operon, while the X-gal is cleaved by beta-galactosidase to turn E. coli cells blue.
And now for question 3. So it gives us this long DNA molecule. We didn't write the whole thing. It's pretty long, but it says write the single-stranded DNA sequences that you would use for the 2 primers used in the PCR experiment. And so we went ahead and wrote those on the board here. So this is the reverse sequence and the forward sequence. And so the reverse sequence will attach to, let's see, the reverse sequence will go 5' to 3' on the DNA, and then the forward sequence will go 3' to 5', but you can look at it either way, and it's still the primer that you need.
So here's
Try to move it closer. Oh, I guess I'm destroying everything. There we go.
Okay, so for part B, it gives you a mathematical equation to find the annealing temperature of each of the primers. And so the equation is 81.5 plus 0.41 times the percentage of your C and G in each primer, and then minus 65 over the number of nucleotides in those primers. So for the forward reaction, we get 39.5 degrees Celsius, and for the reverse, it's going to be the exact same. Okay.
There we go. So for part C, this works. So for part C, it says what annealing temperature would you predict for each primer from part A above? And so sorry? Oh, sorry, yeah, I didn't read the whole question. So each PCR cycle contains a DNA synthesis step, and then it asks what the sequences of the 2 newly synthesized strands of DDCTP, the 2 newly synthesized strands are if DDCTP is accidentally included in the PCR experiment. And so I wrote it here under C. And these are the 2 strands. And so as you see, there's only 1 cytosine in each one. And this is the case if DDCTP is accidentally included in the PCR experiment. That's the question to see, or answer to see.
Okay. Part D says, it talks about DNA replication and how we learned about DNA-A and deamethylase. And it asks why these 2 proteins are not included in the PCR experiment. And that's just because we don't really need them. We're going to have the heat that's going to anneal everything. And I believe the deamethylase is part of translation, which is not what we're even trying to do.
Mm-hmm. So for question 4, part A, thank you. It gives us a bunch of different restriction enzymes and the DNA fragments that they produce. And so for part A, it says to draw an image that represents a result of agarose gel electrophoresis. And so Kristen has this nifty drawing here. And so as you see, the heavier elements that are 1,000 base pairs are not going to go nearly as far as the ones that are 100 base pairs long. And they each have different fragments. So for example, BAM H1, they go down to 50, 350, and oops, I'm pointing on the right side. Yeah, so 50, 350, and 500 base pairs. And so that's illustrated here. And so on and so forth. And that's how you draw agarose gel electrophoresis.
Okay. For part B, it asks us to draw a linear restriction map. And this is what ours looks like. And we just took the combined restriction enzymes and their fragments, and just tried to figure out where they fit with each other.
Yeah, and then for the last one, using that map, we were able to find which kinds of sections there are. And so there's a 50 section, another 50 section, a 100 section, 200 section, then a 500 section. And so those are the ones that we got for part C.
Okay, and then for part 5, it talks about a yeast species, and it asks you to tell the steps that you would use to generate cDNA from this yeast to a mature mRNA cell, clone cDNA, and then use the blue-white screening. So for part 5, so for the first part, we're going to have to purify the mRNA, and then you'll mix it with a primer. And then after that, you're going to use reverse transcriptase and GNTPs to synthesize the DNA. And then you add an RNase to digest the mRNA. Then you add DNA polymerase 1 to synthesize a second strand. And then I just added at the end that plasmids will be back, and that's going to disrupt everything. So thanks for watching.
Yeah, those are our answers. Have a great day.